Okay, good day. And our topic is all about the DNA fingerprinting. And I will introduce to you the key points of our report. Discuss what is DNA fingerprinting or DNA profiling. Explain the stages of DNA profiling and differentiate the uses of DNA profiling, crime between medical problems. Discuss the types of the DNA fingerprinting and explain the process of DNA fingerprinting. And discuss the biological materials used for DNA fingerprinting. And last, discuss the advantage and disadvantages of DNA fingerprinting. And let us proceed to the what is DNA profiling? A technique used by scientists to distinguish between individuals of the same species using only samples of their DNA. The only difference between people or any animal is the order of the base pair. DNA fingerprinting, another term for the genetic fingerprinting. In other words, DNA profiling is the process of determining an individual's DNA characteristic called DNA profile. This is a test to identify and evaluate the genetic information called the DNA. Now, who invented it? The process of DNA fingerprinting was invented by Alec Jeffreys at the University of Leicester in 1985. He was knighted in 1994. And these are the stages of DNA profiling. The stage 1, cells are broken down to release DNA. If only a small amount of DNA is available, it can be amplified using the polymerase chain reaction or PCR. So the isolation of the DNA. DNA must be recovered from cell tissues of the body. Only a small amount of tissues like hairs, blood, skin needed. And the stage 2 is the DNA is to cut into fragments using restriction enzymes. Each restriction enzyme cuts DNA at a specific base sequence. Cutting, slicing, and sorting. Special enzymes called restriction enzymes are used to cut the DNA at specific sites. The DNA pieces are sorted according to size by sharing technique called electrophoresis. And the stage 3 is the fragments are separated on the basis of size using a process called gel electrophoresis. DNA fragments are injected into wells and an electric current is applied along the gel. So the transfer of DNA to nylon by placing the sheet on the gel and soaking them overnight. So next is proving. Adding and radioactive colored probes to the nylon sheet produces a pattern called DNA fingerprint. So the DNA fingerprint is to build by using several probes, 5 to 10 more simultaneously. So the stage 4 is the pattern of fragment distribution is then analyzed. So this is the uses of DNA profiling. DNA profiling is used to solve crimes and medical problems. Crime forensic science is the use of scientific knowledge in legal situation. The DNA profile of each individual is highly specific. The chances of two people having exactly the same DNA profile is 30,000 million to 1 except for identical twins. So there are may only be a few drops of blood or a single have left at the scene of a crime, but that is enough is to get a DNA STR profile. Forensic scientists can compare the STR profile of a blood sample with that of a suspect or the victim to look for a match. If two DNA profiles match, there is only 1 in a billion chance that they are from two different people unless they are identical twins. So next is the DNA profiling can solve crimes. The pattern of the DNA profile is then compared with those of the victim and the suspect. If the profile matches the suspect, it provides strong evidence that the suspect was present at the crime scene. It does not prove they committed the crime, and if the profile doesn't match the suspect, then the suspect may be eliminated from the inquiry. So next is the solving medical problems. The DNA profiles can be used to determine whether a particular person is the parent of a child. A child paternity is the father and maternity is the mother, and it can be determined. This information can be used in paternity suits, inheritance case, and immigration cases. So for example, paternity test by comparing the DNA profile of a mother and her child, it is possible to identify the DNA fragments in the child which are absent from the mother and must be therefore have been inherited from the biological father. And next reporter will be Miss Sheina D. Quinanola.
Okay, good day. So I will be discussing the types of DNA fingerprinting methods. And those are electrophoresis, polymerase chain reaction or PCR, and last is the restriction fragment length polymorphism or RFLP. Okay, so what is electrophoresis? So electrophoresis is a separation technique that is based on the mobility of ions in an electric field. Positively charged ions migrate towards a negative electrode, and negatively charged ions migrate toward a positive electrode. Ions have different migration rates depending on their total charge, size, and shape, and can be therefore be separated. So electrophoresis came from the Greek word meaning bearing electrons. So electrophoresis of a positively charged particles or cations is called cataphoresis, while electrophoresis of negatively charged particles or anions is called anaphoresis. When we say electrophoresis, it is a technique used in laboratories in order to separate macromolecules based on size. Next is the polymerase chain reaction. So the polymerase chain reaction or PCR was developed by Carey Mullis of the Cetus Corporation in 1988. In this process, the DNA sample is denatured and separate individual strands. Specific DNA primers are used to hybridize to two corresponding nearby sites on DNA strands in such a fashion that the normal enzymatic extension of the active terminal of each primer, that is the 3 plus end, leads toward the other primer. In this fashion, two new copies of the sequence of interest are generated. So the polymerase chain reaction is a technique used in molecular biology to amplify a single base copy or a few copies of a segment of DNA across several or orders of magnitude, generating thousands to millions of copies of a particular DNA sequence. So the next type is the restriction fragment length polymorphism. So the RFLP analyzes the length of the strands of the molecules with repeating base pair patterns. The basic technique for detecting RFLPs involves fragmenting a sample of DNA by a restriction enzyme, which can recognize and cut DNA wherever a specific short sequence occurs in a process known as restriction digest. So the resulting DNA fragments are then separated by length through a process known as agarose gel. So electrophoresis and transferred to a membrane via southern blot procedure. So the restriction fragment length polymorphism is a difference in homologous DNA sequence that can be detected by the presence of fragments of different lengths after digestion of the DNA samples in question with specific restriction endonucleosis. Let's proceed to the process of DNA fingerprinting. So the first one is, example, the blood sample. The next process is DNA is extracted from blood cells. The third one is DNA is cut into fragments by a restriction enzyme. Next, DNA fragments are separated into bands during electrophoresis in an agarose gel. The fifth process is that DNA band pattern in the gel is transferred to a nylon membrane by a technique known as southern blotting. Next, the radioactive DNA probe is prepared. Next is DNA probe binds to specific DNA sequences on the membrane. The eighth process is that exists DNA probe is washed up. And then in the ninth process, at this stage, radioactivity probe is bound to the DNA pattern on the membrane. Next is X-ray film is placed next to the membrane to detect the radioactive pattern. And last is that the X-ray film is developed to make visible the pattern of binds, which is known as DNA fingerprint. Next, let's proceed to the biological materials used for DNA profiling. First is the blood, next is the hair, next is the saliva, semen, body tissue cells, and DNA samples have been obtained from vaginal cells transferred to the outside of a condom during sexual intercourse. So first is the forensic hair analysis can be done to help identify a criminal by evaluating hair structure and DNA from cells attached to the root of their hair. Next is the saliva is the most common DNA sample. And the next is the semen. So DNA test of semen can take several days or several weeks before the results are evaluated depending upon which techniques have been used by the lab. Okay, let's proceed to the advantages of DNA fingerprinting. So the first one is it helps people know more about their family background. So DNA fingerprinting can be used to help a person know who his parents and siblings are. It's incredibly helpful for people who become separated from their family because of adaptation, kidnapping, war, natural calamities, and other causes. It is also helpful for parents who want to know if their child is really theirs or not. So the next advantages is that it helps solve crimes. So DNA fingerprinting can help law enforcers in two ways. It can assist in identifying suspect through the body tissue that they leave on the scene of the crime, such as semen and rape bodies or pieces of skin. So DNA fingerprinting can also help with identifying victims, particularly those whose body becomes so decomposed that they can't identify outright. So next, the third advantages of DNA fingerprinting is that it can help determine and even prevent illnesses. So many people have used DNA fingerprinting to know which illnesses they are predisposed to have. So let's proceed to the disadvantages of DNA fingerprinting. So the first one is it can violate people's privacy. So DNA fingerprinting can be seen as a tool that violates the privacy of people and makes their personal information easily available to others. The second disadvantage is it can point the finger at the wrong person. So there have been cases wherein DNA evidence was planted crime scene at a crime scenes to accuse the wrong person. So the last disadvantages of DNA fingerprinting is that it's complex and prone to errors. So DNA fingerprinting is a complicated and time-consuming procedure that needs sophisticated machines and fully trained lab specialists. So this is the end of our report about the topic of DNA profiling or DNA fingerprinting.